Hello, everyone, and welcome back. In this lecture, we will discuss Chapter 4, which covers cell structure and the endosymbiotic theory. Robert Hooke was an English scientist and architect. His contributions to science cover an array of topics, such as the properties of gravity and the wave-like properties of light. In 1665, he released the first ever publication of microscopic life, which is life that cannot be observed with the unaided eye. To the right, you can see the microscope he used for his drawings in Micrographia. Here are a few of the images he drew from what he observed under the microscope. At the top left is a water flea, and below that is mold. In the center are the veins and the cross section of a stinging nettle plant, and to the right is a drone fly. Probably the most profound image he drew was the mesh-like structure to the right. Here is an image of a cross section of a sample of cork tree, and he noted in his writings the microscopical pores that were the first ever observed. Now, at the time, he was not aware of what these were, but these were the first cells observed. These pores, he identified, were in fact the fundamental unit of life that we call cells. In this image, we can see the dark red stains of the inside of the cells, which are the different stages of the genome throughout cell replication. The genome of a living organism is the entire set of DNA found in its cells. And as you recall, one of the characteristics of living organisms is order. And in this lecture, we will cover the order and structure of cells. And there are a set of tenets to be aware of with cell theory. There are parts of cells that are even smaller, but those parts by themselves are not living, because individually they cannot meet all 8 characteristics of living organisms. These 3 tenets of cell theory are: cells are the smallest living units, all living organisms are composed of cells, and all cells arise from pre-existing cells. All of the images presented here are cells. At the top are plant cells, which have a rigid structure due to the presence of cellulose within their cell walls. The rod-like cells are a type of bacterial cells. If you look carefully, you can see a pair dividing into 2 separate cells. And at the bottom are a type of animal cells. If you note the different coloring inside the cells, again, that is the DNA going through a process of replication called mitosis. And each cell is going through a different stage of mitosis, and that's why the red dye appears to be in different shapes or scattered at different times. We're going to cover some commonalities of all cells at first, and then talk about structures that differentiate some cells from others. One structure that all cells share is the plasma membrane. This membrane is what allows cells to be distinguished from their surroundings. This barrier is incredibly thin and helps regulate what can enter or exit a cell. It is made up of what is called the phospholipid bilayer, as you can see in the images here. It is called a bilayer because it has 2 layers of phospholipids: an outer layer whose phosphate heads face outwards, and an inner layer whose phosphate heads face towards the inside of the cell. It does this because the phosphate heads are polar and therefore attracted to water through hydrogen bonding. The inside of the cells are mostly made of water and chemicals suspended in it, and the outside world is also generally wet. Therefore, the phospholipid layers each naturally align themselves accordingly. Additionally, the fatty acids of the phospholipid do not like water because they are nonpolar, but are attracted to themselves, so they automatically align inward. The nature of compounds that are repelled by water are called hydrophobic, coming from the word for water hydro and for fear phobic. Philic means loving, so hydrophilic compounds are attracted to water. These associations occur spontaneously. If a beaker of water is combined with phospholipids, A layer of phospholipids would quickly form at the top, with all of the hydrophilic phosphate heads pointing inward and hydrophobic fatty acid tails pointing upward. Cells are spherical, however, and so to account for the inside and outside, a bilayer will automatically assimilate. Additionally, all cells have a semi-fluid substance inside the cell. This semi-fluid is a solution of water packed with many different molecules that make it almost a gel-like substance. A water-based solution, just so you know, is called an aqueous solution. So this solution the cytoplasm would be called an aqueous solution. This substance is called the cytoplasm, and cyto means cell. There also must be genetic material, which for living organisms is a set of DNA that encodes the characteristics used and passed down to its offspring, and ribosomes, which are clusters of proteins and RNA. And these ribosomes take the information from DNA and use it to synthesize the cell's proteins. All organisms in the world are made of either prokaryotic cells or eukaryotic cells. They are characterized one of each category depending on whether or not they have a nucleus. A nucleus is an area of a cell that houses the genome, or DNA, of the cell. Like the cell itself, it is surrounded by its own envelope of membrane. The name karyo means nucleus, and eu means true. Therefore, eukaryotes are organisms that have nucleuses. Prokaryotic cells are the most genetically diverse and predate eukaryotic cells. They do not have a nucleus, but rather a what's called nucleoid region or nucleus-like region, which is a region where the DNA is concentrated but not stored within a membrane. And so you're aware, pro means pre. So therefore, prokaryotes evolved pre-nucleus. Prokaryotic cells are small and seemingly simple. As previously mentioned, they have no nucleus, but they also have no internal membrane-bound structures. They have never evolved to become multicellular. Some examples you may be familiar with are Escherichia coli or E. coli, which is a bacteria found in all of our guts, and Streptococcus pyogenes, which is found on our skin, or Lactobacillus, which is principally involved in yogurt and cheese making. They can come in many different shapes, but one of the most common shape to a prokaryote is the pill shape. We shall cover some of the key components of prokaryotic cells, starting with the nucleoid, which is an area of the cell that the genetic material is found. The flagellum extends from the cell in long strands. It is made of protein, and at the base is a motor that spins the filament, causing the cell to propel. The flagellum allows prokaryotes to be mobile, and this is called motility. The cell wall is a special outer layer of the prokaryotic cell. It is made of a substance called peptidoglycan. Peptides are just small proteins, and the root word glyco and gluco means sweet, as you may have heard of glucose, glycemic index, or glycogen, and these words are all related to sugar. Therefore, peptidoglycan is a structure made of proteins and sugars. Peptidoglycan is a rigid material that gives the cell wall extra rigidity and strength, and thereby helps protect the cell and maintain its shape. The capsule is a fine layer made of carbohydrates outside the cell wall that provides further protection to the cell. Checkpoint 1. The antibiotic penicillin interferes with the compound peptidoglycan. Which structure in a prokaryotic bacterial cell does it destroy? The capsule, DNA, membrane, or the cell wall. Next, we have the eukaryotes. Eukaryotic cells are typically much larger than bacteria. On average, they are at least 10 times the size of bacteria. They have many internal structures called organelles, which are surrounded by their own membrane, and they can be single-celled, but many have also evolved the ability to be multicellular. Therefore, if you can imagine a multicellular organism, such as mushrooms, elephants, and humans, we are all going to be eukaryotes. Intuitively, it feels like there is so much more to us because of our multicellularity. And the prokaryotes look like little insignificant blobs under a microscope. While we are most intimately familiar with other eukaryotes, and that's most of what we see in the visible world, if we take a step back, eukaryotes take up much less of the biodiversity on planet Earth. If you look at the image here, we have three branches of the tree of life. Uh, Now, you don't need to be bothered by the details of all the organisms listed on these branches, but be aware that the two main branches are, in fact, prokaryotes, and the eukaryotes make up one of the more minor, smaller branches. Eukaryotes can be but can be broken up into subcategories of plants, animals, fungi, and protists. Protists are the one that we might be less familiar with here, and it's a catch-all for eukaryotes that are none of the first three and are mostly, but not always, found to be single-celled. Some eukaryotes also have a cell wall that helps maintain the shape and protect the cell. Plants and fungi have cell walls, but protists and animals do not. Checkpoint 2. Which is not a component of cell theory? A. Cells are the smallest living units. B. All living organisms are composed of cells. C. All cells have cell walls. Or D. All cells arise from pre existing cells. The dyed areas inside these cells are their nucleuses. They are surrounded by a double layered membrane called the nuclear envelope, and the nucleus is the site within nucleolus is the site within the nucleus where the ribosomes are synthesized. And as you recall, the ribosomes take the information from DNA to synthesize proteins. The endoplasmic reticulum, abbreviated as ER, are made of a single continuous membrane inside the cell, folded up many times upon itself. The rough endoplasmic reticulum is studded with ribosomes, which are the site of protein synthesis. The smooth ER lacks these ribosomes and therefore appears smooth. It contains membrane associated enzymes that synthesize lipids and steroid hormones. The Golgi apparatus are a series of sacs of membranes material within which proteins and lipids from the endoplasmic reticulum are modified chemically. There are also regions where cell wall materials are synthesized and molecules are modified and discharged in vesicles. Vesicles are small spheres of membrane material that bud off organelles to transport materials. Remember the term hydrolysis, which means to break water, with lysis and hydro? Well, lysosomes are a subset of vesicles that contain digestive enzymes for breaking down large particles. The cytoskeleton is a mesh of protein fibers that runs throughout the cell, It gives support to the shape of the cell, keeps organelles fixed to their locations, and are used as railways to transport material around the cells. The vacuole is a sac filled with water and stored materials. Vacuoles are not found in animals. Their materials can be used for a variety of purposes. For example, the central vacuole in plants stores water and allows cells to regulate their shape and size. And the contractile vacuole, which can be seen in this GIF here to the right, can pump water in and out and therefore regulates the amount of water in the cell and protists in response to environmental conditions outside of the cell. Checkpoint 3, match the structure to its function. 1. Rough ER. 2. Vacuole. 3. Golgi Apparatus. 4. Smooth ER. A. Protein modification and vesicle packaging. B. Protein synthesis. C. Storage of water and materials. D. Lipid and steroid hormone synthesis. Chloroplasts are organelles found in all plants that are responsible for photosynthesis. Photosynthesis is the reaction that uses photons, in other words, light, to synthesize biomolecules, importantly, sugar. Interestingly, chloroplasts contain their own DNA other than that found in the nucleus of plants, which indicates something very special we'll talk about in a moment here. Mitochondria are the region of a very important process we'll cover in more detail later called cellular respiration. Cellular respiration is a set of chemical reactions that breaks down sugar to obtain energy for the cell. They are found in the vast majority of eukaryotes and also contain their own DNA, just like chloroplasts do. Checkpoint 4. Compare and contrast the structure of prokaryotic and eukaryotic cells by describing two structures they have in common and then two structures that they do not have in common. Checkpoint 5. Name three structures that plant cells have that animal cells do not. What are their functions? There is an elegant explanation as to why mitochondria and chloroplasts contain their own DNA, and it is called the endosymbiosis theory. It states that chloroplasts and mitochondria evolved from ancestor prokaryotes that were engulfed from another ancestral prokaryote and became intracellular residents that over time became the organelles we know today. Just take a moment to reflect that within your own very own cells lives bacterial ancestors. And remember, this is not the same as E. coli in your gut. E. coli do not live inside of your cells. They merely live inside your intestine or your GI tract. Here we're talking about cells living within cells, in a sense. Although we don't call them cells, we refer to them as organelles. And it seems far fetched, but there is an incredible amount of evidence to support this theory, as are all theories, and some of which are the characteristics that they share with prokaryotes. For example, they have a membrane bilayer. And while other organelles have membranes as well, these ones contain their very similar structures in the membranes as found in prokaryotes. And they also have their own set of DNA. And if you were to study them genetically, they like a paternity test, their genes are most closely related to prokaryotes and not the eukaryotic hosts. They also have their own ribosomes for making their own proteins. And again, these ribosomes, their structure, their amino acid sequence, is most similar to prokaryotic ribosomes, not eukaryotic ribosomes, which indicates their ancestry is was derived from prokaryotic ancestors. Additionally, they are about the same size and shape as prokaryotic cells. Now, keep in mind, as I said earlier, eukaryotic cells are about 10 times the size on average than prokaryotic cells. So there's plenty of room inside of those cells to carry these prokaryotic cells. And they replicate through binary fission. We haven't gotten into replication yet, but binary fission is the process of how bacteria multiply, and it's a matter of splitting into two equally sized cells from their parental cell. The image shown here is a living organism that still exists today that was recently discovered. It's called Margarita lithophora, and it's a photosynthetic bacterium called a cyanobacterium. And cyanobacterium are thought to be the ancestors of the chloroplasts. Coincidentally enough, they both synthesize, they both do photosynthesis, and their genes are most closely related. In this particular organism right here, its gene sets are the most closely related to the genes found in the chloroplast DNA of any other living organism on Earth. And in case the theory still doesn't seem quite plausible to you, or you just can't accept the concept that you have little bacteria like organelles floating in all of your cells, we can look at nature today and find the process of endosymbiosis actively taking place in many different organisms. In this figure, each one of these cells has cyanobacteria, not organelles, but cyanobacteria that are living transiently or temporarily inside and then outside of them. And someday, just like chloroplasts and mitochondria have, they may take a permanent residence in their descendants. Scientists have recently found a correlation between the size of the genome or the DNA sequences in an endosymbiont and the time period in which their endosymbiosis event occurred. So let me explain. On this graph, you will find the genome size on the x-axis. So the bigger the genome size, the longer their DNA is. And on the y-axis, you'll find the number of protein coding genes. And what scientists have discovered is that the as endosymbionts go from being an intracellular bacterium to evolving to becoming an organelle, over time, some of those genes that they have become redundant and not necessary to keep up with. So they lose those genes. And so the older a organelle is, as far as like historically when it was first just a intracellular bacterium, the fewer genes it has. And so the first endosymbiont we have in eukaryotes, basically all eukaryotes share it, is the mitochondria. And if we look at this graph, we can see mitochondria have a very small genome and they have much less protein coding genes than the bacterial genomes in blue, which is in the uh, top right corner, where they have the most protein coding genes and the biggest genome. So what that tells us is that over time, the endosymbiont loses the ability to do many tasks. Now, it was first the mitochondria, then the what they call plastids here. You can think of those as the chloroplasts, which are in orange. Those were the next ones to come along. So plants have both mitochondria and chloroplasts. And so first they became eukaryotes with the mitochondria. Then they gained the cyanobacteria that later became a chloroplast as it sort of devolved and lost some of its genes and figures and traits and became the chloroplast. And another area where evolution is actively taking place and forming new endosymbionts are the insect endosymbionts, which are listed here in pink. And so they're kind of sort of transitioning here from bacterial genomes to these organelles. And if you are a gardener, you may be familiar with mealybugs. And here on this potato are mealybugs. There are little white spots here shown with that arrow. And mealybugs are interesting because they get all of their nutrition by sucking the juices from plants. Well, unfortunately, it's not a way for the mealybugs to get all of their necessary amino acids. And so mealybugs have acquired an endosymbiont that's capable of converting some of those amino acids to other amino acids so they can get all of their essentials. And what's so fascinating about this relationship is that endosymbiont, that little organelle, that little bacterium that lives inside of the mealybugs, well, it lost the ability to make its peptidoglycan. And eventually, nature decided that it was best if it could still make its own peptidoglycan for whatever reason. And so it has garnered its own endosymbiont. So we're looking at a sort of a Russian nesting doll situation where you have a eukaryote that has its mitochondria, of course, but it also has another endosymbiont that makes amino acids for it, which has its own endosymbiont, which makes peptidoglycan for it. Incredible. And with our organelles, we can get diseases that are associated with malfunctioning organelles, such as the Golgi apparatus. Um, this is achondrogenesis, which is a disease characterized by a lack of normal bone and cartilage development, and it's usually fatal before or after birth. And that is the end of Lecture 4.